Chapter 3, Section 3.5, Nucleic acids. Nucleic acids are polymers of nucleotides with very specific functions in cells. DNA is the genetic material that stores information regarding its own replication in the order in which amino acids are to be joined to make a protein. RNA is another type of nucleic acid. One type of RNA molecule called messenger is an intermediary in the process of protein synthesis, conveying information from DNA regarding the amino acid sequence in a protein. Some nucleotides have independent metabolic functions in cells. For example, some are components of enzymes, non-protein organic molecules that facilitate enzymatic reactions. ATP is a nucleotide that supplies energy for synthetic reactions and for various other energy requiring processes in cells. Structure of DNA and RNA. Every nucleotide is a molecular complex of three types of molecules: phosphate, a pentose sugar, and a nitrogen-containing base. In DNA, the pentose sugar is deoxyribose, and in RNA, the pentose sugar is ribose. A difference in the structure of these 5 carbon sugars accounts for their respective names because deoxyribose lacks an oxygen atom found in ribose. There are 4 types of nucleotides in DNA and 4 types of nucleotides in RNA. The base of a nucleotide can be pyrimidine with a single ring or purine with a double ring. In DNA, the pyrimidine bases are cytosine and thymine. In RNA, the pyrimidine bases are cytosine and uracil. In both DNA and RNA, the purine bases are adenine or guanine. These molecules are called bases because their presence raises the pH of a solution. Nucleotides join a definite sequence by a series of dehydration reactions with DNA and RNA form. The polynucleotide is a linear molecule called a strand in which the backbone is made up of a series of sugar-phosphate molecules. The bases project to one side of the backbone since the nucleotides occur in the definite order. So do the bases. RNA is a single-stranded. Right. In section 3.5, I learned about nucleic acids, the structure of DNA and RNA, and how every nucleotide is a molecular complex of 3 types of molecules. Here's a picture of nitrogen-containing bases, and here's another picture of phosphate, nitrogen-containing base, pentose sugar, deoxyribose versus ribose, and ribose.
DNA is double-stranded with the 2 strands usually twisted about each other in a form of a double helix. The 2 strands are held together by hydrogen bonds with pyrimidine and purine bases. The bases can be in any order with a strand, but between strands, thymine, as always paired with adenine, and guanine is always paired with cytosine. This is called complementary base pairing. Therefore, regardless of the order or the quantity of any particular base pair, the number of purine bases always equals the number of pyrimidine bases. Table 3.3 summarizes differences between DNA and RNA. ATP. ATP is a nucleotide in which adenosine is composed of adenine and ribose. Triphosphate stands for the 3 phosphate groups that are attached together into ribose, the pentose sugar. ATP is a high-energy molecule because the last 2 phosphate bonds are unstable and are easily broken. In cells, the terminal phosphate bond is usually hydrolyzed to give the molecule ADP and phosphate molecule.
Okay, so today I learned about the chemistry of organic molecules. DNA is a double-stranded structure that is formed in a double helix. The 2 strands are held together by hydrogen bonds between pyrimidine and purine bases. The bases can be in any order, but they have to be in their pairs, which is thymine always goes with adenine and guanine is always paired with cytosine. Those are called complementary base pairing. ATP is a nucleotide in which the adenosine is composed of adenine and ribose. The triphosphate stands for the three phosphate groups that are attached together to the ribose and pentose sugar. The ATP is high in energy molecule because the last two phosphate bonds are unstable and are easily broken.
The energy that is released by ATP breakdown is coupled to energy requiring processes in cells. For example, the energy required for the synthesis of macromolecules, such as carbohydrates and proteins, is derived from ATP breakdown. ATP also supplies the energy from muscle contraction and nerve impulse conduction. Just as you spend money when you pay for a product or a service, cells spend ATP when they need something. Therefore, ATP is called the energy currency of cells, because energy is released when the last phosphate bond of ATP is hydrolyzed. It is sometimes called the high energy bond, symbolized by a wavy line. But this terminology is misleading. The breakdown of ATP releases energy because the products of hydrolysis are more stable than the original reactant, ATP. It is the entire molecule that releases energy, not a particular bond. In my part, I learned about adenosine and triphosphate, which is also ATP. And ADP is the adenosine with the diphosphate and the phosphate plus energy. And right here, you can see the two together, the adenosine and the triphosphate put together.